So good morning everyone. This is Dr. Shyma. We continue with visual pathway today. So in last class we had in detail discussed about the functional anatomy of the eye. So each and every component of the eye, including the receptors, the pigments, everything was discussed in the previous class. Now before I actually move on to the visual pathway, I would want you all to know a little about, you can just have a look at the picture, the response of basically the receptors which are present in the eye to light. You know that the neurons, or you call them as the specially structured neurons, which are present in the eye are the rods and the cones. And these specially structured neurons, the rods and the cones, they act as receptor for the light stimulus which is reaching the eye in the form of photons. When light in the form of photons touches these receptors, the rods and the cones, they convert the light energy into chemical energy that gets transmitted to the bipolar cells as synaptic potential. Now, there are 4 different photosensitive compounds which are present in the retina, 1 in rods which we call as a rhodopsin and 3 in cones. Now, this picture will actually tell you about, the response of these specially structured neurons when light falls on them in the form of photons. Now have a look at the first picture which tells us the response, what happens in dark, and the second part of the figure tells us what happens in light. Let's see the first picture. In dark, the sodium ions are transported into the outer segment through a channel which you call as the cyclic GMP mediated sodium channels, and that causes hypopolarization. You can see that hypopolarization word being given at the end of the picture. Now when this happens, so I just repeat the sentence once again. In the dark, sodium ions would make an entry into the receptor through cyclic GMP mediated channels, which will cause the hypopolarization of the receptor. Now when hypopolarization of the receptor happens, you can see at the synapse there is release of the neurotransmitter glutamate at the synaptic knob of the rods. Now when this neurotransmitter glutamate, there is a persistent release, it will cause a persistent hypopolarization of the bipolar cells. This is what is happening in dark. Now look at the next picture. I think we'll continue here. So when there is a persistent release of glutamate at the synaptic knobs of the rods, which is causing a persistent hypopolarization of the bipolar cells, when the bipolar cells are continuously hypopolarized, this will inactivate the retinal cells. So when the retinal cells are inactivated, there is no impulse being conducted along the visual pathway. Just keep that in mind. Visual pathway, I'll take you all after few slides. Now look at the second picture. You can see that light is activating there the G protein transducin which is present in the receptor membrane. Now when the G protein transducin is activated, this will indirectly activate phosphodiesterase which destroys cyclic GMP. When cyclic GMP is destroyed, sodium channels close, which results in hyperpolarizing generator potential in the synaptic terminal of the photoreceptors. Now what happens to the release of neurotransmitter glutamate? That is going to drastically come down. Now the rods and the cones do not generate action potentials, but instead they generate graded hyperpolarization potential, which depends on the intensity of the stimulus, the amount of light which is falling on them. The rod receptor potential now has a very sharp onset and a slow offset potential. In contrast, you can see that the cone receptor potential has a very slow onset and a sharp offset potential. So now whatever I have explained to you all, regarding this figure, I have put all of them in the form of bullets in the coming up slides. So which would actually help you all to make your notes. I think you can directly follow it. The points have been, briefly condensed. The most important points have been put down. Now let's have a look at this picture. You can see that, this is basically two functional pathways which is connecting the primary visual cortex, which is area number 17, and their functions are also described. So you have it with, on the left side the rods and the cones, and on the right side you can see what happens with cones. So when the impulse from the rods and the cones through the magnoganglion cells, it will reach the first and the second layers of the lateral geniculate nucleus, magnocellular, and it reaches the 4C alpha of area 17. And basically the impulse coming through that pathway, the functions are, it gives a rough sketch of the object, more faster response, and basically the perception of black and white objects happens here, and even the detection of movement happens through the sensation coming through that pathway. Now if you look at cones, the impulse coming from the cones picked up by the parvoganglion cells through the 3, 4, 5 and 6th layer, the parvocellular layer, it reaches the 4A and 4C beta of area 17, and that helps in the perception of finer details of the object, and it's concerned with accurate vision, but the response is slow, and the most important thing is that takes care of the color vision. Now we go to the visual pathway. Now if you look at this picture, you can see that you can see few blue fibers and also red fibers. And the blue fibers, we call them as the temporal field fibers, and the red fibers are the nasal field fibers. So I think as I explain, you can follow the visual pathway, so there's a better understanding. So visual sensations, arise in the rods and the cones, and whatever sensations which are arising in the rods and the cones are transmitted to the bipolar cells that conveys the sensation to the ganglion cells. Now from the retina, the fibers of the ganglion cells congregate to form the optic nerve as they come out through the optic disc. Now the optic nerve travels to the optic chiasm, where about half the fibers of each eye cross over to the opposite side. After the optic chiasma, the fibers travel as the optic tract to the lateral geniculate body. Now the fibers from the temporal half of the retina, that means the blue fibers, they do not cross the midline and travel in the optic tract of the same side. That means the red fibers are the temporal field fibers. I think I've made a small mistake at the beginning. The red fibers are the fibers which are taking care of the temporal field of vision, and the blue colored fibers are taking care of the nasal field of vision. So the nasal fibers actually cross at the optic chiasma. I'll slightly enlarge the figure so you can have a more better view. So you can see that the blue fibers which are taking care of the nasal field of vision are crossing over, whereas the red fibers which takes care of the temporal field of vision remain on the same side. They are ipsilateral all through the pathway. So I just repeated. Now the fibers from the temporal half of the retina do not cross the midline and travel in the optic tract of the same side. Now the blue fibers from the nasal half of the retina, they decussate at the chiasma and travel in the optic tract of the opposite side. Thus each optic tract, just see where we use the term optic tract to what combination of fibers. Each optic tract contains fibers arising from the temporal half of the retina of the same side and the nasal half of the retina on the opposite side. Now the direct and the crossed fibers which are present in the optic tract pass to the alternating lamina of the lateral geniculate body and it ends there. Can you see there? The lateral geniculate body, it ends there. Now, from the lateral geniculate body, again axon, the neuron arises and it projects to ipsilateral primary visual cortex. To be specific, it's Brodmann's area number 17, which is located on the sides of the Callicrine fissure on the medial aspect of the occipital lobe of the brain. Now, the fibers carrying the visual impulse from the lateral geniculate body to the Callicrine cortex are called the geniculocalcarine tract. You can see that being named in the figure. Now, as these fibers spread out from the lateral geniculate body, they radiate. So, they are also known as the optic radiation. So, that's about the visual pathway. Now, the optic nerve fibers, listen very carefully here, they project to several other areas of the brain like the superior colliculus, pretectal nuclei, association visual cortex and the suprachiasmatic nuclei of the hypothalamus. That's not actually shown in the figure here. Here you can see only a projection to the visual cortex, Brodmann's area number 17. Now, these fibers which have these projections to the superior colliculi, pretectal nuclei, etc., they actually participate in regulation of function which are associated with vision. So, when visual pathway is asked, you need to draw this figure, and how the pathway goes exactly the way I have explained. You need to pen down the points, and to make it convenient for you all, I have actually put all the points in bullets. You can just have a note of it. So, now I take you all to the next portion, which is basically the effects of lesion at various levels of the visual pathway. Can you see a black line? I'll just slightly enlarge this. Can you see there at the optic nerve, a black color marked, which you need to read it as a lesion A, which is a lesion of the optic nerve of the right side. Now, you can see lesion B, which is actually a lesion at the optic chiasma. Now, lesion C, which is lesion of the optic tract of the right side. Now, lesion D, which is a cut or a lesion of the geniculocalcarine tract. So, let's see what happens when these lesions happens in the body. So, you can see that in this picture on the right hand side, the left visual field and the right visual field is being mentioned here. So, now A, let's see at lesion A, which is a lesion at the optic nerve. So, lesion of the right optic nerve produces blindness of the right eye, isn't it? Because both the nasal field of vision and the temporal field of vision is lost, because optic nerve, it's the fibers from the same side, because the fibers haven't crossed yet. Now, lesion B, lesion of the optic chiasma, produces bitemporal hemianopia. So, I think okay, I think I'll just continue with that explanation and might be tell you more on what hemianopia is, what you exactly mean by bitemporal hemianopia. I'll explain it in the coming up slides. Now, lesion C, where the lesion occurs at the optic tract, it produces left homonymous hemianopia. Now, just remember the word. With this figure again, I'll explain you all what it is. Now, lesion D, when the lesion is at a geniculocalcarine tract, it produces left homonymous hemianopia with something called as a macular sparing. Can you see at the center? So, now I think you can just have a look at this picture. I'll explain you what exactly the hemianopia means. So, now blindness of the visual field, you use the term anopia. Now, hemianopia means half blindedness, which can be either homonymous or heteronymous. What do you mean by homonymous? Same side of both visual fields are affected. Now, the lesion of the optic nerve, that means I am talking about lesion A here. It actually cuts off impulse transmission from that particular eye and produces blindness in that visual field. That's why with lesion A here in this picture, you can see blindness of the right eye. Now come to the lesion B, where the lesion of the optic chiasma damages the nasal fibers from both the eyes, isn't it? The blue fibers are cut here and produces bitemporal hemianopia. And this condition usually occurs in tumors of the anterior pituitary, in which the enlarged pituitary presses upon the optic chiasma and produces this bitemporal hemianopia, a feature of acromegaly. Now lesion C, where the lesion is at the right optic tract. So lesion of the right optic tract damages ipsilateral temporal fibers and contralateral nasal fibers, which is resulting in homonymous hemianopia. Now lesion of the left optic tract produces right homonymous hemianopia. Please don't get confused there. And lesion of the right optic tract produces left homonymous hemianopia. Now coming to lesion D, which is a lesion of the geniculocalcarine tract. It damages the ipsilateral temporal fibers and contralateral nasal fibers, resulting in homonymous hemianopia with macular sparing. Why does that macular sparing happen? The macular fibers travel dorsal to the optic radiation, and that's why they escape damage. The visual field defects are similar to that of the optic tract. Now one more situation can happen where that's not marked in the figure if lesion occurs at the occipital cortex. Sorry. Now if lesion occurs at the occipital cortex, it damages the ipsilateral temporal fibers and contralateral nasal fibers with macular sparing. It produces scotoma in the homonymous visual fields. And what do you mean by scotoma? Scotomas are blind patches. So I think we are done with the visual pathway and the effect of lesion at the different levels of visual pathway. So I think whatever you need to write, I have actually put down in the PPTs. So now we go to, I had made a mention about the fibers which radiates to, other visual areas. So I think you would, this can come as a 3-marker for you all, so you should have a brief understanding about it. Now the primary visual cortex is, I've already mentioned, it's Brodmann's area number 17. It receives sensory information regarding vision. So basically whatever the vision you have, the impulses needs to reach the Brodmann's area 17. Now visual cortex or Brodmann's area 17, it receives information regarding movement, depth, velocity, color of an object, and it also helps in the depth perception and stereopsis, or you call it as the binocular depth perception. Now the second slide tells us about which are the visual association areas. Area 18 and 19 are the visual association areas, and they are located on the lateral part of the occipital cortex, and they receive information from the visual cortex which are concerned with interpretation of the visual stimulus. Now any finer attribute of an object, what you see, it can be a texture, location, orientation object, everything you will be able to know if the impulse reaches this visual association areas. Now lesion of the visual association areas can produce impairment of these higher visual functions. Now the frontal eye field. The frontal eye field, which is area number 8, is present in the frontal gyrus, and it is responsible for the eye movements. The superior colliculi, it receives input from the optic tract fibers and also the visual cortex, and it coordinates eye movements like saccade. Saccade is basically, the rapid movement of the eye between, fixed 2 points, the convergence, and it basically keeps the eyeball in focus. Now you have the pretectal nucleus. The fibers which arise from the optic tract gives branches to the pretectal nuclei, and through these branches it activates the Edinger-Vespa nucleus. and they are responsible for the pupillary reflex, which are required for the regulation of visual fixation. Suprachiasmatic nucleus, it's a small nucleus in the medial hypothalamus. It's located just above the optic chiasma. It receives fibers from the optic nerve and it's concerned with the regulation of circadian rhythms. So now we come to the errors of refraction or the defects of the image forming mechanism. Now I think I would just enlarge the picture. So you can see the first picture. So basically the refractive condition of a normal eye is known as emmetropia, and the eye is known as an emmetropic eye. Now in an emmetropic eye, the parallel rays of light from a distant object are focused on the retina without accommodation. Now if the refractive state differs from the normal, then we call it as emmetropia. Now in emmetropia, the parallel rays are focused in front of the retina or behind the retina, resulting in a blurred image. Now if this condition occurs in children, one of the images is suppressed, causing loss of vision in one eye. This is called amblyopia or anopsia. Now emmetropia is of three types: myopia, hypermetropia and astigmatism. Now the refractive errors of the eye may be due to abnormality of the following reasons: either the axial length of the eye, the refractive power. Under refractive power, you can put 3 points: curvature of the surface of the cornea or the lens, the refractive indices of the media, or the position of the lens. So in the picture, the first part, you can see the emmetropic eye, which is a normal eye where the light rays are falling exactly on the retina. Now the second picture, we discuss the first defect in the image forming mechanism, which is myopia. We also call it as the short-sightedness. Now myopia, it's basically a refractive error in which the parallel rays of the light from a distant object are brought into focus in front of the retina. Now the light rays in an emmetropic eye, which was falling on the retina, here in myopia is falling in front of the retina. Now the cause of myopia either can be acquired or it can be genetic. Now congenitally the eyeball may be elongated, or that means the anterior posterior diameter of the eye is increased. Myopia is acquired. How is it acquired? When there is increased duration of close works over years, something like reading, etc. Which continuously, when a person is doing that, it will activate the accommodation process, especially the lens becomes more convex. Myopia is normally corrected by a biconcave lens. Now when you use a biconcave lens, it will converge the biconcave lens will diverge the incident rays before it strikes the cornea. Now look at the second picture. So you can see the first picture, myopia, you can see that the light rays is falling in front of the lens, where when you keep a biconcave lens in front, the light rays falls on the, so basically you can tell that myopia is corrected by a biconcave lens. Now come to the second picture. You can see hypermetropia or farsightedness being discussed there. Now hypermetropia is a refractive error in which the parallel rays of light from a distant object are brought into focus behind the retina. Now when the accommodation is at rest, I think that only that completes the definition of hypermetropia. It occurs mainly due to decreased anterior posterior diameter of the eye, and it's normally corrected by using spectacles with biconvex lens, and now that will converge the incident rays before they strike the cornea. Now the near point is further than normal, so the patient needs the biconvex lens at an earlier age. You can see that's very clearly mentioned in the figure. A convex lens is used and hypermetropia is corrected. Now you have two more remaining, astigmatism and or also presbyopia. Now if you look at astigmatism, this is basically a refractive error. I think I have the slides coming up here. Yeah, it's here. So astigmatism, this is what happens. It's a refractive error in which the parallel rays of the light from a distant object cannot converge to a point for focus on the retina. Now what is the reason? Why can't the light rays converge to a point focus on the retina? Due to unequal refraction at different meridians. Now because the reason is the corneal surface is not perfectly spherical. So what happens is the curvature of one median differs from the other. Accordingly, the light rays are focused at different points on the retina and which will cause a blurring of the image. It can also occur if the lens curvature is not uniform or if the lens is pushed out of alignment, and astigmatism is corrected by a cylindrical lens. Now let's come to presbyopia. Now presbyopia, basically this is a physiological phenomenon seen in all persons at about the age of 40 years, in which the near point of vision comes down beyond the normal reading or working distance, and this happens due to the progressive loss of plasticity of the lens. Now as the near point recedes beyond a point at which one is used to do close work or read ordinary prints, which is normally around 28 centimeters from the eye, the person will have difficulty in reading, writing, or kind of when you try to perform any kind of close work. And presbyopia is corrected by convex lens for near works, usually prescribed in the form of a bifocal lenses, so that when the person wants to change his focus from a near to distant object, each time he need not take off the glass. So that's how what presbyopia is and how presbyopia is corrected. So I think there we come to the end of visual pathway with the effect of lesion at different levels of visual pathway. The refractive errors are also discussed, where we talked about emetropia, myopia, hypermetropia, astigmatism and presbyopia. Initially I had given you all an introduction of exactly how the neurotransmitter is released and how when light falls on this modified neuronal cells, the rods and the cones in the eye, how the light energy is getting transported into chemical energy and action potential. It travels along the visual cortex. It falls on Brodmann's area number 17, visual cortex, and how the person is able to see things around us. So there we finish with today's class. I think next class I would, there are a few more things to be done when it comes to vision. So hopefully next class I would wind up with vision. So thank you. Thanks for listening, guys.